Welcome. Today, we're going to dive into a brand new, groundbreaking map of the human genome. It's a tool that's completely changing how we understand the very instructions that build and run our bodies.
All right, let's just jump right in with a really big question. Think about it. Every single cell in your body, whether it's in your brain or your liver, has the exact same DNA, the same genetic blueprint. So, what's the deal? How on earth does one cell know to become a neuron, while another one becomes a liver cell?
Well, to answer that, you have to think about our DNA a little differently. It's not just some long string of code. It's really more like a huge, incredibly detailed instruction manual for, well, you.
And this instruction manual, your genome, is inside almost every single one of the trillions of cells that make you who you are. It has all the instructions needed for literally everything your body does.
But here's the secret sauce. No cell reads the entire book from cover to cover. Instead, it's like each cell type gets its own personalized copy with specific parts highlighted. A brain cell highlights all the chapters on being a neuron. A liver cell highlights the sections on liver function. These biological highlighter marks are what scientists call chromatin states, and they're the master switches that tell each cell what it's supposed to be.
So you can probably guess that figuring out these highlighter marks is a huge deal in biology right now. I mean, if we can map them all, we can understand precisely how our cells work, how they specialize, and crucially, what happens when they go wrong.
And that brings us to this absolutely massive scientific project called ENCODE. It stands for the Encyclopedia of DNA Elements. For years, thousands of scientists have been collaborating on this one giant mission to create a complete catalog of every functional part of our genome, including all those super important highlighter marks.
And what's really cool is how scientists have developed a whole language to describe these highlights. You can see here they've got about 11 key terms. For instance, a promoter is basically a mark that says, hey, start reading the gene right here. An enhancer can act like a volume knob, cranking up the activity of a nearby gene. And then you have quiescent, which is basically the genome's way of saying, nothing to see here, folks, move along.
Now, very recently, the ENCODE project released a new version of this map, and it is incredibly detailed. It was built with a much more powerful technique called Segway, and it gives us a view of our genome's activity that's just so much sharper than anything we've had before.
And the scale of this thing is, well, it's just immense. We are talking about super detailed maps for 234 different types of human cells and tissues. And all of that data was pulled from almost 1,800 different experiments. It's hands down the most complete map of its kind ever made.
So how in the world do they make these maps? Well, it's this really clever three-step process. First, the Segway software scans the genome and chops it up into segments that have similar patterns of activity. Then, and this is the cool part, a machine learning classifier automatically figures out what those patterns actually mean. And bam, out comes the final product, a complete, high-res map for a specific cell type.
And this slide really gets to the heart of why Segway is such a game changer. You see, older methods were kind of like a simple on-off light switch. A gene was either active or it wasn't. But Segway is like a dimmer switch. It doesn't just see on, it sees how on it is. It measures the intensity of the signal, which means it's sensitive enough to pick up on all these subtle but totally critical details we were missing before.
Okay, so we've got this super sensitive method, but how does the system actually translate what it's seeing? I mean, trying to make sense of thousands of patterns and hundreds of different cell types, that is a monster of a challenge.
Trying to interpret all this data by hand would be, well, it would be basically impossible. So the researchers came up with this brilliant semi-automated system. They painstakingly, manually interpreted a small set of these genetic highlights. Then, they used that small, perfect set to train a machine learning model. And that model just took off, accurately interpreting thousands and thousands of other patterns all by itself.
So, you have to ask, how good is this automated system really? Well, this chart, which is called a confusion matrix, is basically the model's report card. And that strong, bright line you see going right down the diagonal, that tells us it's an A+ student. It almost never gets confused. It knows the difference between a promoter and a totally inactive quiescent region. And that level of accuracy is absolutely essential if scientists are going to trust and use this map for their own research.
And this is where things get really personal. Because this isn't just some abstract science project. Let's look at how these incredible new maps are actually helping us understand human diseases.
For a long time, scientists have used this tool called GWAS. It lets them scan the DNA of thousands of people to
to find tiny little variations that are linked to diseases like diabetes or heart disease. The only problem is a GWAS study can tell you that a variant is linked to a disease, but it usually can't tell you where in the body that little variant is actually doing its damage.
This is where the new map completely changes the game. It works like a GPS for our genome. So when a GWAS study finds a genetic variant linked to a disease, researchers can now overlay that onto the ENCODE4 map and see exactly which of the 234 cell types that piece of DNA is active in. It connects the what of the disease to the where.
Let's make this real. Researchers took genetic variants that we know are associated with coronary artery disease, and they checked them against the map. And what did they see? Well, just look. The activity for those variants lit up right where you'd hope to see it, in a bunch of different heart tissues. It's a huge confirmation that the map is working just as it should.
Okay, now let's zoom all the way out. This heat map shows you the true power of this catalog across tons of different diseases and traits. Every one of those colored squares is a significant link between a genetic trait, anything from bipolar disorder to insomnia, and a specific cell type where that genetic switch is being flipped. This is an absolute treasure trove of new biological connections that researchers are just now starting to dig into.
So, this new encyclopedia of our genome's master switches really is a massive leap forward. It's an absolutely essential resource that's going to be fueling discoveries about our health for years and years to come, giving scientists a guide as they tackle the next big mysteries of human disease.